Good morning, everyone. My name is Jordan Fantuzzi. I'm a biotech associate here at Piper Jaffray. Our next presenting company is Synlogic. And before I begin, I must draw your attention to disclosures regarding the relationship between Piper Jaffray and Synlogic at the back of the room in registration. So Synlogic is a really cool story. The company is applying synthetic biology in medicine to design a novel class of living medicines that perform specific therapeutic functions. Their lead drug, SYNB1618, treats phenylketonuria in an earlier stage pipeline advances, both in immuno-oncology and GI therapies. So here to present for Synlogic is Dr. Aoife Brennan, President and CEO. Aoife, thank you for joining us.
Thanks, Jordan. It's great to be here.
So I want to start high level. I mentioned Synlogic is pioneering the use of synthetic biology to develop these synthetic biotic medicines. So tell us, what are synthetic biotics and how are you using synthetic biology to engineer them?
Yeah, so it's always a pleasure to tell the Synlogic story. The company came together really around two kind of really exciting and breaking areas of science. On the one hand, we had this kind of increasing realization that the bacteria that live in and on us and around us have a big, profound impact on health and disease. And there were a lot of companies pursuing, a microbiome approach, an ecology approach to try and unlock some of that potential. And we were really excited by that science, but thought if there was a way that we can be more precise and mechanistic, that could be a really interesting platform. And around the same time, we became aware of the potential of synthetic biology based on our accumulating knowledge of sequencing and our ability to print DNA and our ability to control functionality with specific switches and promoters. So basically bringing these two worlds, these two areas of science together, we founded a company called Synlogic, and the core of the company is using a probiotic bacteria that's been very well studied and characterized called E. coli Nissl, understanding, what mechanism is really driving a particular disease state, and then engineering that mechanism into the probiotic chassis so that you intercept or prevent disease. So that's kind of the concept behind the company. And as we started to think about how we would translate this really interesting science into a pipeline of products that have the potential to really help patients with specific diseases, we realized that there were a couple of key components that we wanted to understand and to really establish the kind of foundational elements of our platform around the regulatory path, the synthetic biology tools, our ability to manufacture, our ability to deliver these to particular sites of action within the human body. And that's basically been the basis of the work that we've done over the past 2 or 3 years to really understand and to build that foundation that will underpin all of our programs, be they internal programs or programs that we partner with others. So that's kind of the background at the high level.
So I think that's a good place to start, and maybe we can get a little more specific using your lead drug, SYNB1618, as an example to describe the synthetic bio workflow in the clinical application. So SYNB1618 is in the clinic to treat phenylketonuria, or PKU, which is caused by these excess production of phenylalanine in the body. So really help us understand how SYNB1618 manages excess phenylalanine in patients, and maybe how is this mechanism unique to this class of drug as a living medicine?
Yeah. So PKU is an ideal first program for us to think about applying our technology to, because it's a disease of high unmet need. The biology is very well understood. These patients have a defect in the enzyme and the gene that metabolizes phenylalanine into tyrosine. So they get this toxic accumulation of Phe in their blood, and if that's not detected early in life, these patients can have irreversible cognitive impairment. So we knew that the bad actor was Phe, and we understood that in order to, treat the disease, we could start to work engineering a probiotic bacteria that consumed phenylalanine. So the first thing, once you have that kind of what you need to do kind of piece sorted out, the next thing we start to do is to think about how are the ways that we can design a bacteria to consume phenylalanine. So we look into the genome of our starting organism, which is called E. coli Nissl, It's a probiotic bacteria that's been used for about 100 years as a health benefit. For health benefits, it's available in Europe and Canada, so we know a lot about it. And we've also established a lot of tools that allow us to tweak the genome of E. coli Nissl. So the first thing we do is we look and see, are there genes that are already in the genome of E. coli Nissl that could consume or could break down phenylalanine in the GI tract of patients with PKU? Well, it turns out that there aren't. So then we look throughout the kind of bacterial universe, and we look at what are the other enzymes and genes that might be beneficial in allowing our strain to metabolize Phe. So we identified a gene that we thought would be interesting. We plug it into the genome of E. coli Nissl, and then we start our iterative design process. And it's a core tenet of synthetic biology, these iterative design circles, where you design, build, test, you evaluate how well you're doing, and then you go back to the beginning again. And we did a couple of cycles there and realized, okay, let's move now into some preclinical models. We're starting to see some in vitro, we're starting to see that these bacteria are consuming Phe. They're consuming Phe at a substantial rate. Let's start to move into some animal models. So we did a bunch of animal work in preclinical models of PKU, where we saw that the bacteria within the GI tract was able to pull down systemic phenylalanine in these PKU animal models. So that gives a lot of encouragement. But it's a big step from a mouse to a human, so we started to do some non-human primate work. We were also able to identify significant activity of the bacteria in the GI tract of non-human primates. And then we moved into the clinic, initially into healthy volunteers with a very early formulation of our product. It was a liquid formulation that needed to be stored at -80. We realized that this wasn't going to be viable long-term as a commercial product, but we really took that kind of concept of design, build, test to heart in terms of how we think about, iterating and designing these programs, and thought, well, if we can get some early clinical experience, at least we'll know how we can tweak and continue to improve and evolve. So we did a phase 1 study with the liquid formulation. We had the nice thing about our PKU program is we have this really nice quantitative biomarker. So we were able to identify that the engineering circuits that we had engineered into E. coli Nissl were actually functioning within the human GI tract. So that was kind of a stunning achievement for us, to be able to see that, in vivo in the human GI tract, that we were able to design a bacteria to perform a specific function, put it into humans, and measure that the bacteria was indeed performing the function that was designed. There was also some really nice safety data that we got from that trial, showing that the bacteria don't colonize the GI tract long term. They behave very well. As soon as you stop taking them, within a couple of days, they're cleared from the GI tract of patients. And some of those things were really important for us as we started to think about our platform and how we might, meet regulatory requirements around follow-up and safety and some of those other important elements. So we did the phase 1 healthy volunteer study. We saw this nice dose response, beautiful PK and PD data around the activity of the bacteria. And then we had the question of, well, is it active enough? Where do we need to get to in order to have our clinically meaningful benefit in patients with PKU? So we've started now down that path of translating, our activity biomarkers into something that's really clinically meaningful. And we realized that, over the long term, this liquid formulation wasn't really going to fly. So we needed to transition to something that was stable at room temperature refrigeration conditions. So we invested a lot of time and effort to establish manufacturing infrastructure, learning, of course, from all of the prior platform companies in gene therapy and in other areas where this was kind of a key differentiator. Getting manufacturing right early on in development, I think, has really been key. And so we invested very heavily in that area over the past 12 months. We established internal manufacturing capability, spent a lot of time developing quality assays, and are now back in the clinic with the solid oral form of the PKU product, where it has nice stability. It starts to look like something that could be commercialized one day. We're currently evaluating that for safety and activity in a healthy volunteer cohort. If that looks good, we'll move on to PKU patients to evaluate the impact on blood Phe lowering. And I think that could be a really interesting program for us, where certainly we'll learn a tremendous amount and hopefully be able to deliver something that's really meaningful for patients with PKU.
No, the program has advanced so much over the last year, and so it's encouraging to see that you've that you've observed translation of the preclinical effect into now humans with respect to Phe lowering. And I kind of want to pick up on some of the data that you saw in the phase 1 with respect to Phe lowering, which I understand has allowed you to create a predictive model of Phe lowering, and you know, what percentage, based on this model, what percentage of Phe lowering is predicted to translate into clinical benefit in PKU patients? And also maybe you can speak to how this has informed your dose selection going forward as well.
Yeah, absolutely. I'm personally a very big fan of model-based drug development, where you start from where you start thinking about what's going to be in your label, right? What's going to really be meaningful for patients, and then you work backwards from that, and you're kind of always trying to maintain line of sight to that eventual product profile. So kind of going through that calculus in reverse order, if you will, based on a lot of conversations with PKU patients, parents of PKU kids, we felt that a Phe lowering of about 30% was going to be really meaningful in terms of a treatment advance. Obviously, we'd love to be able to achieve more than that, but that was kind of what we set as our minimum product profile. And then we started to work out, well, how do we've seen some nice biomarker data. We know that the bacteria are consuming phenylalanine in the GI tract. How can we use the data that we have to calculate whether or not we're in the ballpark of being able to achieve that? And so we looked in the literature, and it turns out that back in the 90s, a metabolic physician had made a model that relates dietary Phe restriction with blood Phe levels. So we thought, okay, this could be a really interesting tool for us. If you believe the assumption that taking 1 mole of Phe out of the diet is the same as having our bacteria consume a mole of Phe, which to us seemed like a reasonable assumption, then we can start to relate the amount of biomarker we're seeing to what we might expect to see in terms of Phe lowering. And so we built this model. We evaluated it a couple of ways, qualitatively and quantitatively. We went around to some dietitians and said, does this kind of seem to make sense with your experience of dealing with patients with PKU and their dietary modifications? And they felt, yeah, that's very interesting. It seems consistent with my experience. And then we started to plug in our biomarker data into the model. And that showed us that in order to achieve our 30% target, we needed to get the dose of the bacteria up a little bit. So we need to be in the E11 kind of range in order to achieve the 30% Phe lowering. So right now we're planning to answer 2 questions. Number 1 is, can we get to that range with the solid oral material? And once we see that we can get to that range in healthy volunteers, we'll proceed with the Phe lowering study in patients with PKU, which will really answer the question of how good is our model at predicting Phe lowering. And we think that will be, very interesting data, and will really allow us to think, very logically about how we think about, backup programs, moving this program forward, formulation changes. So I think it's a really interesting tool for us, because as a small company, we can't afford to do a big phase 3 trial that fails. Right. So the more information, the more de-risking we can do before making that big investment, the better. And, I'm a big believer in modeling and simulation, and really trying to de-risk as much as possible before you make that big investment on a registrational trial. So that's kind of where we are right now.
Yeah, I think that makes a lot of sense. And so right now you said you were studying the oral formulation of SynB in this bridging study to see if you can reach the adequate dose level in healthy volunteers. How is that study going, and do you plan to report data from that study?
Yeah, absolutely.
Yeah.
So the study will be complete before the end of the year, and we'll be reporting data shortly thereafter. And if that looks good and we think we have a dose that we can really take forward to validate our model, we'll be initiating that Phe lowering study in the first half of next year.
And going back to the first healthy volunteer study, had you seen any sort of safety or tolerability issues with the doses that you had tested?
Yeah. So across, we've done a couple of trials now in patients, or in in humans, I should say, and it seems that we don't see any systemic toxicity. We don't see any long-term colonization. The dose-limiting toxicity that we see is GI upset, which I don't think is unusual or unexpected based on our kind of class, our class of drug. So what we see when we start to reach the maximum tolerated dose, patients get nausea and vomiting after dosing. So that's really the big AE that we manage. If patients continue through, continue with dosing, generally it kind of subsides over a period of time. But that's the kind of the big safety or the tolerability issue that we've seen so far. Nothing in terms of infections or any systemic toxicity that would be worrying over time. But yeah, that's been how we've...
Okay. Well, we'll look for those data with the study completing by year end to kind of inform the next steps for SYNB 1618 in the phase 2 PKU study, which will be a big milestone for the company. I want to shift gears now. You had a clinical setback earlier this year with SYNB 1020 that was in the clinic for cirrhotic patients. Mm-hmm. With SYNB 1618, I think we discussed a very linear, kind of predictive, thoughtful approach to drug discovery. And I want to kind of understand what you learned from the 1020 setback that's going to help you design more successful synthetic biotics in the future.
Yeah, I think the SYNB 1020 was our first program. I think we learned a tremendous amount. It was the program that kind of defined us as a clinical stage company. We built a lot of our clinical and regulatory experience off that program. And every program has its strengths and its weaknesses. I think some of the weaknesses in the 1020 program was we had a biomarker of strain activity, but it wasn't quantitative. It was pursuing ammonia lowering, which I think has been a challenging field generally, just because I think the biology of ammonia and the relationship between the GI tract and ammonia lowering is less clear compared to in PKU. And also it has a more challenging forward-looking development path in the two indications that we had considered as potential indications for the strain. So I think while we learned a lot and we're disappointed to see that the preclinical data didn't predict what we saw in the clinic, in some ways we were kind of, it allowed us to focus on things where we really feel there's a much clearer kind of biological pathway and there's better validation between diet and disease activity, if you know what I mean. It really wasn't there for the ammonia program. So
yeah, I think that makes a lot of sense and is pretty understandable. So picking up on that relationship between metabolite scavenging and GI tract, what are some other metabolic indications where you could see this pipeline going after PKU?
Yeah, so I mean, the vision for the company is that we would focus, we will have to continue to build our foundational components in terms of the underpinnings of our platform. And that will feed a pipeline of programs, some of which will be internal programs and some of which will be programs that we will pursue in the context of a partnership. Ideally, as a company with all the platform risk, we want to take internal programs that we believe have very well-validated biology, where we really understand how much activity we need, where we can really create that model and do a calculation of how much of this substrate do our bacteria need to consume in vivo, and have good certainty that if our bacteria do that, the likelihood that it will translate to a clinically meaningful endpoint is very strong. Because I think that from a risk profile perspective, doing all the platform novelty and all of the investment we've made in really understanding our platform means that we have to choose straightforward biology for our internal investments and pipeline. And we think that there are a number of really interesting diseases with high unmet needs, where the biology is well understood, where there's an identified toxic metabolite that builds up that could be a really good application for an engineered biotic approach. And we've been working very hard to kind of tee up those next programs internally based on the learnings from 1020 and from the PKU program. But we also see this platform as something that's not limited to just the kind of toxemias, if you will. And we think that there are a lot of really interesting applications that could leverage the technology that we've built. And often those applications are in diseases and areas where there's a bigger commercial opportunity, but where there's also some biology risk. Diseases like inflammatory bowel disease and oncology. I think there's enough information in the academic literature now, certainly for me to believe that there is a really good relationship between bacteria, the microbiome, and cancer, and bacteria and inflammatory bowel disease. The key question is, what are the mechanisms? And how are we going to identify those targets that are really driving the disease pathology? And that's an area where we think, sharing some of the risk with a big pharma partner could be really interesting for us, right? Where they believe that there's a specific target that has good biology that they're interested in investing in. They need a partner who can deliver that with a new technology that's not really amenable to a small molecule or biologics approach. And those are the ideal partnerships for us, where we can continue to strengthen our platform, but share some of the biology and target risk for a potentially bigger indication over time.
Mm-hmm. Yeah, so maybe we'll start there. Let's focus on shift gears from GI to the immuno-oncology side of your platform. I think there's a very real, at least preliminary understanding that there's a relationship between the microbiome and cancer, but how are synthetic biotics uniquely suited for this task to modulate the immune system?
Yeah, so I think there are 10 different ways that bacteria can interact with oncology.
Mm-hmm.
There are things from, the microbiome as a way of predicting response to checkpoint inhibitors. There's there are 10 different approaches. Our strategy was based on the belief that the innate immune response is going to be important in immuno-oncology. And the thought process was, our immune systems have evolved to recognize bacteria as a trigger for innate immune activation. And when we looked across the spectrum of the various kind of innate immune activators that were out there, they were all kind of causing almost like a fake bacterial infection. TLR, Sting, these are all things that happen as part of bacterial infection. We also realized that there'd been a number of attempts to use bacteria in oncology historically, and a lot of them failed because of safety issues. They were pathogenic strains that were attenuated, or using BCG as a way to stimulate the innate immune response. And we were really inspired by the toolkit that we have from a synthetic biology perspective. So here we are, we can take this historic observation but bring it into the kind of 21st century with synthetic biology tools that allow us to engineer specific functionality into the bacteria and take out some of the safety risks and vulnerabilities with the bacterial platform. So we thought this is too good an opportunity for us to pass up on. We thought that the best way to really understand whether this was a real opportunity for our platform was to pursue a strategy that was based on the best combination of innate immune activation that we could identify to start a trial. And we've done just that. We've engineered a non-pathogenic strain of bacteria that expresses a STING agonist. We've shown preclinically that it stimulates multiple different pathways of the innate immune response. We've shown that it has a very differentiated profile compared to, say, a small molecule naked STING agonist. Mm-hmm. And we've now moved forward with a clinical trial where we're injecting the bacteria directly into the tumors of patients who have failed to respond to all available therapeutics. And we'll have some data on that approach next year. So I think it's a really exciting opportunity, very different to the oral programs. But I think if we can really understand the potential of engineered bacteria to deliver effectors into the tumor microenvironment, it could really stimulate a pipeline of programs with engineering based on various effectors that might be important in specific tumor types. So we thought that was absolutely an experiment worth running, and we'll learn a lot from that in the clinic in the next 12 months.
Yeah, I think the thought behind that program is really clear and will definitely be a significant kind of proof of concept if you're able to show positive data for the STING agonist next year, so we'll look for that. So we have just about a minute remaining, and in that time I want to talk about some of the partnerships that Synlogic has signed, specifically with AbbVie for inflammatory bowel diseases and most recently the Ginkgo collaboration. Both super cool in and of itself. So maybe you could just kind of give us an overview on where those partnerships are at.
Yeah, absolutely. So the AbbVie partnership was one that we did very early in our kind of life as a company, and was really based around that principle that we discussed earlier of we're not the experts in IBD. We want to be the experts in our platform and partner with big pharma partners who have targets that they believe are interesting from a therapeutic perspective in specific disease states that we then engineer into our biotics and advance through preclinical development. So AbbVie is a great example of that. They've been in the IBD space for a long time. They're the leaders in this area. They have a tremendous research infrastructure looking at new targets and validating new targets. They brought us a number of targets, and we've been executing and designing engineered bacteria to deliver those targets in the GI tract. So that's been a great example of the kind of partnership that we'd like to do more of in terms of really helping strengthen the platform. Ginkgo is a very different type of partnership. We realized that there was so much happening in synthetic biology that we'd love to have access to, but it was going to be really hard for us to build internally with our focus on therapeutic development. And so Ginkgo was a great, it gave us the ability to access multiple different technology in a very high throughput way that they've built over the past 10 years at Ginkgo in a way that was very targeted. So it's been a phenomenal technology partnership for us. It's been very successful, and it's been a great experience since we signed the partnership about 6 months ago.
Great. Well, lots coming down the pipeline. Aoife, thank you.